Okay, so my name is Cindy Cron. If you haven't met me before, I'm the North Coast IPM advisor. I cover Napa, Sonoma, Lake and Mendocino counties. And today we're going to talk about a new insect in pear fruit, and it's an update from presentations that we gave last year. So I will cover, if you've already seen my presentations from last year, I will cover the same information, and then I'll move into what we learned this year. Just so that you know, not everyone online might have heard all the details from last year. So I want to make sure that everyone's up to date of where we stand currently with this insect. So in August of 2023, we received photos from Brock Zoller from Pear Orchards in Lake County, showing on the photo on the left-hand side, you can see a black mark on the pear fruit, and near the calyx also a smaller black mark. And so when he cut back that piece of fruit to see what was causing that damage, there was a flat-headed borer larvae that you can see in the photo on the right-hand side, in a little pocket full of frass, which is their poop, and a very dry environment that was created inside of the pear. So we started looking out in the field, and you could find a wide range of damage out in the field. And so you can see just from a tiny little spot on the left-hand side to, that spot getting larger to a very large portion of the fruit having the black spot on the sunny side of the fruit. And so here's another photo showing variation of what I could find in the field of this damage. And so by seeing this in the field, I knew that there was a flat-headed borer, at least one inside. We did find up to 5 larvae per fruit. Sometimes there was one, sometimes a few. It ranged. And the damage also ranged in the fruit that we saw in the field. So here's a couple more photos of what I was finding in the field when I was walking the orchards. On the left-hand side, you can see 2 fruits of varying degree of damage, and then 1 fruit on the right-hand side. Again, these black spots always occurred on the sunny side of the fruit, so they were on the side of the fruit that was facing out. So it was very easy to see the damage. It was not hidden under, facing towards the center of the tree. It was facing outward at all times. So one of the things is showing the difference between codling moth damage, which most people are familiar with, which is on the right-hand side, and compared to this flat-headed borer damage. It's similar, but it's not the same. Codling moth damage, you can see on the right-hand side, has frass, the brown. It's basically their poop that they push out of the fruit as they feed. And so you can always see frass on the outside, the brown frass. Where the flat-headed borer damage, the frass remains on the inside of the fruit. So you see the blackened portion near the calyx and near the sunny side of the fruit, but you don't see any external frass near the entrance or exit holes. So that's one of the biggest differences between the damage out in the field that you could be able to use to tell the difference between codling moth and flat-headed borer damage. So where have we found the flat-headed borers in pear fruits? In 2023, we found the larvae in 4 pear orchards in Lake County, but it should be kept in mind that this discovery was late in the season, and area-wide scouting was not possible to determine the extent of the distribution before harvest. So we found it not long before harvest. We did some searching in orchards, but there was not enough time to do a wide area-wide search to see, is this very localized? Is this throughout, the whole county? How often did we find it? So all these things the questions were not able to be answered because the discovery was right before harvest. Sorry about that. So as an entomologist, flat-headed borer in fruit was just mind-boggling. And when I shared this information with other entomologists, they're like, wait, what? In the fruit? Are you sure? Because when you think of the classification of this insect, so we have phylum, class, order. The order is Coleoptera, which is beetles, and that's all beetles. And we have 400,000 plus species of beetles in the world. This family, which is a smaller classification of beetles, it's a type of beetle, is Buprestidae, and their common name is metallic wood-boring beetles. So this is a beetle known to be wood-boring, and of the 15,500 plus species, when we searched the literature, there was no documentation in the literature of any of these species attacking fruits of any kind. So it was very mind-boggling that we would see this behavior. It would not be a behavior that we would expect. And so uh, it was definitely when we first found her, like, wait, this doesn't make any sense. um, but we sent off a sample to CDFA, and they were able to identify to Chrysobothris genus, but we were not able to get a species identification. The reason being is that the larva on the right-hand side is pretty nondescript. It is they don't have the features needed to identify the species, to differentiate one species from another. They're pretty nondescript in general, and you really need an adult to be able to identify what species you have. So in the Chrysobothris genus, we have 690 plus species, of which 69 are known to be in California. So we were slowly narrowing it down to a smaller group when we're looking at 69 species in California. Um, Early on, there was always the question of, well, is this a new invasive species? So maybe this is not a species we normally have in California, which could explain odd behavior. We will see in later slides that that is not the case. This is not an invasive species. So this is a species that naturally occurs in the US. So that's something to keep in mind. So in the quest to find our species, I spoke with Jalendra Rajal. He's an IPM advisor in the Central Valley that works with flat-headed borer on walnut. And he connected me with some researchers. First, I talked with Carla Adesso. We got an artificial diet recipe. So we wanted to rear out the larvae to adults, but we had to take them out of the pear fruits and put them on a diet that would sustain them, that they would be able to continue their life cycle, but we could control that environment a little bit more. So you can see the photos on the right-hand side. This was my desk. And each larvae was dissected out and placed on the artificial diet that I created. And we had grow lights on them to be able to mimic daytime, nighttime, diurnal shifts, which are important for insect development. And so we sent that sample off to CDFA. We did receive a Chrysobothris ID but not a species ID. We sent a sample off for DNA analysis to William Klingman's lab, who's a professor at the University of Tennessee. Their lab works extensively with different species of flat-headed borers. And so I waited till about early November, and then I placed these larvae that I had on my desk in the refrigerator for 10 to 12 weeks. And the purpose of this was early November would be a time in which the environment out in the field would start to change. Well, it would have already started to change prior, but it's really you having your shorter days. It's a lot cooler. And so some insects need to have that cooling temperature in order to complete their life cycle. And so we placed the larvae in the fridge to mimic winter, to help them move along and be able to pupate and then emerge as an adult. So this process at that time could take 5+ months, and it really did. So to get an adult, and the reason being is if you look at the generalized life cycle of a flat-headed borer, we will find adults in June and July. They lay their eggs at that time, and then the larvae emerge starting in July. and they started to burrow, normally burrow into wood, but in this case, they were burrowing into the fruits of the pears. And you can see that majority of their life cycle, they have one generation a year, majority of their life cycle, they're larvae, right? They're larvae from July to the following May, June kind of time. And so um, we we intercepted these in July. I'm sorry, in August. So you can see very early on in the life cycle, we had the larvae. So we kept them until November, if you continue on looking at the graph, and then put them in the fridge for 10 to 12 weeks, and then brought them back out and put them on a heating pad and external lights to be able to get them to come out of dormancy, out of their winter time, and hopefully, in theory, pupate. So in the meantime, we sent off larvae to William Klingman's lab. And so we dissected, you'll see on the top left, larvae out of pear fruits that were infested, that we collected in Kelseyville. And then I sent a sample of the larvae, preserved in 95% ethanol, and sent that off to their lab in Tennessee. They extracted DNA from the larvae, and they show you photos. PCR was completed using customized COX-1 primers. These COX-1 is the area of the genome that we look at when we're trying to identify one species from another. And so a lot of this goes more into technical detail than what we need. But the point is that they extracted that DNA. They were able to look at a section of the DNA that we use to identify species. And take that information, that sequencing information, and run it against a database called the GenBank database, in which other researchers have entered in those sequences and known the identification of what species the sequences are related to. And what we found was that based on the database's resources, it was determined that the DNA extracted from the larvae of the pear fruit was Chrysobothus malai, which is the Pacific flat-headed borer. Now, there's one caveat that early on of why we still needed to rear out adults is that we were basing our identification off of what was in GenBank in a database. And the researchers did not recognize the researcher that entered in the original data. So we were basing it off of whether that information was correct or not. And that's something to keep in mind. There's always a chance that the person that entered it was incorrect. And since there wasn't a way of verifying that, we still needed to rear out an adult to identify using taxonomic keys that could help us make sure that what we have really was Chrysobothus malai, which is Pacific flat-headed borer. So February 29th, very exciting day. I got my first pupa, and it was very exciting because I came into work and it's on my desk, and here we are. One of the larvae pupated, and this is what the pupa looked like. The dorsal side, and so the front and the back, you can see both sides. I flipped it over. And so we were one step closer to getting an adult, which was very exciting. It took a long time and a lot of work to be able to get the larva to this point. And so these are some photos I took under the microscope of the development of the pupa. And as you can see, the exoskeleton starts to darken, and flat-headed borers are really beautiful under a microscope because they get this very iridescent sheen and metallic colors. As you can see on the far right-hand side, it became very metallic green with yellow and bits of pink and orange, and so very colorful insects under magnification. So to the naked eye, you don't usually see this kind of detail. But here we are, March 6th, very close to emerging. And then on March 7th, when I came into work, you can see the photo on the left-hand side, we got our first adult to emerge. We had our second, I think, adult emerge on March 18th. We ended up with 8 adults that emerged, which was pretty good out of 42 that I had on diet. And so we were able to send the first adult off to William Klingman's lab at the University of Tennessee, and they were able to use taxonomic keys to verify that indeed we are dealing with Pacific flat-headed borer. So Pacific flat-headed borer, it is native to the US. It is known to attack apple, pear, peach, apricot, plum, prune, cherry, walnut, as well as other trees and shrubs. And I did write down separate. Some are like maple, oak, willow, elm, poplar, sycamore, beech. So a wide variety of trees. But when I say attack, I mean attack the wood, not the fruit. So larvae will kill or weaken the tree by girdling the trunks and the lower branches of those trees. Newly planted nursery stock, drought-stressed trees, sunburned trees, and trees whose trunks are exposed to the sun usually are the most seriously affected. So this insect has been around. It is native, but this behavior is very different. Why is it going after fruit? It would seem like not really it doesn't make a whole lot of sense. And that's where we came up with the question isn't pear fruit a dead-end host? Because if you think about it, the larvae don't pupate until the following spring. But your pears are not on your tree the following spring. They are long rotten and gone. So the question is, well, okay, is it possible for the larva to complete a life cycle in a pear fruit? And so you can see the photos here that we have here. And what I noticed is that when we would have larvae that were pretty large, and it had been in the pear fruit for a long time, you can see that black spot on the left-hand side is pretty large. So there's some things that I noticed in dissecting them out of pear fruit is when you tap on that dark spot this late into an infestation, that outer portion is really hard. You can hear it. It sounds like you're tapping on wood. And so the outer portion gets really hard. And when I opened up that portion, pulling away from the rotting fruit on the backside, you found that the larvae were tucked away in this little pocket that they create inside. And in that pocket is full of frass, and it's really dry. And if you look at the photo in the middle, you can see the top portion where it's really dry area. And then under it's a very moist, rotting fruit. So they create this environment that is a very dry environment within a fruit that you wouldn't expect. And you can see the photos on the left-hand side. Again, you can really see the dry area compared to the rotting portion of the fruit on the back side. And so also what they create, you can see on the left-hand side, the flat-headed borer sticking its head out, is these right above its head, you'll see this really thin layer of pear that's like leathery. So not only do they create this very dry environment, then there's layers of pear that they tunnel around that makes the pear fruit itself very leathery and dry. So in a way, it creates this little pocket. And in theory, we were thinking, well, it's possible that that pocket can remain dry and protective throughout the season to be able to possibly emerge come early summertime the following year, that you would think the pear fruit would fall to the ground, and it's possible in theory. But we didn't really know for sure. And so Clepson took 9 infested pear fruits and he placed them in plastic jars with a cut on the sides and sealed with a screen. And this helped facilitate the airflow that you would normally have out in an outdoor environment. And this also helped us prevent insects from entering in, but also leaving. So whatever was in that pear fruit, we could be able to see if it's able to complete its life cycle. The jars were placed in a work shed with no temperature control. So we wanted it to be as close as possible to an outdoor environment without it being outdoors. And this was near a window to receive partial sunlight. And the infested pear fruits were monitored weekly from September of 2023 to July of 2024. The ambient temperature that was recorded ranged from 35 degrees Fahrenheit to 103 degrees Fahrenheit. So that's one of the reasons, being in a work shed, it didn't have any kind of insulation to keep it warm or cold. It allowed like normal fluctuations that you would expect, in that environment. And in June of 2024, one adult emerged from 9 infested pear fruits. And so you can see a photo on the left-hand side of what the pear fruit looked like in June, after almost a year of allowing it to dry up and desiccate. And that is the flat-headed borer adult that emerged. And on the right-hand side, you can see the exit hole in which the adult bored out of the fruit and exited from the inside of the fruits. So this did answer the question. Yes, it is possible for Pacific flat-headed borer to complete its life cycle in pear fruits. Now, it's not the same as having the pear fruits outside, but it is the closest that we could do and keeping it a somewhat controlled environment. So remember, we discovered this in 2023, right before harvest. We were not able to do extensive surveys. So in 2024, we conducted visual surveys of presence, absence, and we conducted these on foot in orchards. And basically, we were looking up at the trees, one person looking one side, the other one the other side, and looking at all the fruits as we walk by. Remember that damage, that black spot is on the sunny side, so it'd be facing us, so we would visually be able to see it. And we roughly conducted surveys on 19.3 acres in Upper Lake, 23.8 acres in Scotts Valley, and 69.5 acres in Kelseyville. Part of the reason we conducted in higher acreage in Kelseyville, that is where it was originally found in 2023. So we were trying to sort of look at, okay, is it a centralized area or is it random? And we did have a higher acreage in which we walked in Kelseyville. So in these surveys, you can see the yellow plots. Again, this is a subset, so this is not all the orchards by any means. But we did, at the orchards that we were able to gain access to, and we really tried to gain access to any organic orchards that were available, plus conventional orchards. And these were the orchards that we did, 19.3 acres in Upper Lake, and no flat-headed borer was found this year. These is a map of Scotts Valley, 23.8 acres that we walked, and no flat-headed borer was found. And then when we got to Kelseyville, this is what the maps look like. And we found flat-headed borer in 5 organic orchards. So I wanted to go back. Remember that we had 4 orchards found in 2023. One of those orchards was conventional. And in that conventional orchard, we only found 1 fruit facing the street on the edge tree. So we know that edge trees oftentimes don't get the best application of any kind of pesticide because the rig is turning around. So that could possibly explain why we found it in a conventional orchard. The other 3 were organic orchards. And so in 2024, the orange, you'll see in this map, that orange box, that was the orchard that was conventional that we found in 2023, but we did not find it in 2024. The red ones are the ones that we found in 2024. And so this map is a little bit more zoomed in, showing the 5 blocks that we found them in. So what we found was within, if you were to put a dot in the center of the red orchards, it ended up being a 0.62 mile radius. All the way around, if you were to put a circle, the radius would be 0.62 miles. It's not a very large area. And so they seem pretty centralized and they really seem preferential for these organic orchards. And so, what's next? We did answer some of our questions that we had, and now every time you answer questions, there's now more questions. We just submitted a publication to Pan Pacific entomologists, documenting this new larval host find, and the story of finding a bupestid that's attacking pear fruits. So that is in the process of being reviewed. We are looking at future research questions that we would like to have answered. So we know that the flat-headed borer is being found in a centralized area. And my question at this point is there a change in the genetics in that population that can explain why there's a change in behavior? And the reason why that question is important to answer is that if this is something new, which we don't know, if this is something new and it's a new change in genetics that is changing behavior, then this could, in theory, become a larger problem over time. We have it very centralized now. It's not necessarily going to stay that way. And so that's an important question to look into is there a genetic answer as to why is this? And if there's not, then there's the assumption that this is possibly a behavior that has happened for some time and we are just noticing it. And so there's a lot more diving into these questions to really make sure that are we dealing with a threat that is small that's going to increase over time, or is this something that's always at a low level? So our plan is to continue to share our findings.
No, again.
And we will, as we go into our next year of research, we'll continue to share what we find with the growers to help everyone make the best decisions possible. And with that, I'd like
I
to
made a mistake.
thank the funding provided by the Pear Pest Management Research Fund. These funds are what allowed us to conduct this research. It allowed me to respond quickly and be able to produce the diet to be able to capture the larvae before the pears were gone and harvested, and allowed us to rear them out and get an adult to verify. So having these funds available for the different projects that I do for the pear industry in Lake and Mendocino counties was very important. So thank you, Pear Pest Management Research Fund. And with that, I will conclude my presentation and I can take questions.